Gene editing, the key to saving endangered species? Imagine a world where we can genuinely help endangered species regain their genetic strength. This groundbreaking vision is starting to take shape thanks to advancements in gene editing technology. An international coalition of scientists is showcasing how these innovative techniques, originally developed for agricultural purposes and de-extinction efforts, can be redirected to enhance genetic diversity in species facing extinction. The research team, comprised of conservation geneticists and biotechnologists, is led by Professor Kok van Oosterhout from the University of East Anglia and Dr. Stephen Turner from Colossal Biosciences. Their collaborative effort includes input from notable institutions such as the Colossal Foundation, the University of Kent's Durrell Institute of Conservation and Ecology, the University of Copenhagen's Globe Institute, the Mauritius Wildlife Foundation, and the Mauritius National Parks and Conservation Service, alongside Durrell Wildlife Conservation Trust. We're experiencing the swiftest environmental transformations in history, which pushes many species to the edge of survival due to a critical loss of genetic variation necessary for adaptation, comments Professor van Oosterhout. He suggests that gene engineering methods could act as a lifeline to restore that vital variation. This may involve reintroducing lost DNA from immune system genes sourced from museum specimens or even incorporating genes that promote climate resilience from closely related species. To secure the survival of endangered species, the team strongly advocates for integrating these pioneering technologies with conventional conservation methods. While traditional approaches like captive breeding and habitat conservation aim to increase population sizes, they often overlook the importance of restoring the genetic diversity that disappears when species dwindle. A rebounding population can still be afflicted by limited genetic variation and a high burden of detrimental mutations, a situation termed genomic erosion. Unless addressed proactively, species that have rebounded after a sharp decline may remain vulnerable and less capable of withstanding new threats, such as diseases or climate shifts. Take the example of the pink pigeon, a species that has clawed its way back from near extinction, from a mere 10 individuals to over 600 today, thanks to extensive captive breeding and reintroduction efforts in Mauritius. However, genetic studies conducted by several researchers reveal that despite this recovery, the species continues to suffer from significant genomic erosion, with a risk of extinction looming within the next 50 to 100 years. The pressing task now is to restore the genetic diversity it has lost, a challenge that genome engineering can potentially address. Gene editing isn't a new concept in the agricultural realm. Pest and drought-resistant crops are cultivated on millions of hectares worldwide. Recently, the buzz around plans to resurrect extinct species has underscored just how significant this technology could be. Dr. Beth Shapiro, Chief Science Officer at Colossal Biosciences, asserts, the same technological capabilities that enable us to blend mammoth genes into an elephant's DNA can also be utilized to salvage species on the brink of extinction. It is our obligation to mitigate the extinction threats that currently confront thousands of species. However, they also shed light on potential risks, including unintended genetic modifications and the possibility of inadvertently further diminishing genetic diversity, emphasizing that these new strategies are still in the experimental phase. There's a clear call for careful, small-scale studies and rigorous long-term monitoring regarding both evolutionary and ecological impacts. Engaging local communities, indigenous populations, and the public at large in this conversation is paramount before any extensive application. The authors make it clear that while genetic interventions are promising, they must work hand-in-hand with habitat restoration efforts and other established conservation tactics. As Associate Professor Hernan Morales from the Globe Institute articulates, biodiversity faces unprecedented dangers necessitating equally unprecedented solutions. Genome editing is not a substitute for species protection and will never serve as an all-encompassing remedy. It must be evaluated meticulously alongside conventional conservation methods within a holistic framework that places species protection at its heart. And here's the twist. How do you feel about merging cutting-edge technology with nature conservation? Could this approach signal a new chapter in our fight against extinction? Or does it raise more questions than it answers? Share your thoughts. Can CRISPR save endangered species? Gene editing vs. extinction. Imagine a world where beloved species like the woolly mammoth and the dodo could be brought back from the brink of extinction. This isn't just a fantasy from a Hollywood movie. It's a real possibility thanks to the groundbreaking technology of gene editing. But here's where it gets controversial. While this technology holds immense promise, it also raises significant ethical and practical questions. Let's dive into the fascinating world of gene editing and its potential to save endangered species. Earth's biodiversity is under severe threat. We are on the brink of what scientists call a sixth mass extinction, which could wipe out countless species and drastically reduce genetic diversity within those that remain. This genetic variation is crucial for a species' ability to adapt to environmental changes and resist diseases, making it essential for their long-term survival. Traditional conservation methods, such as protected areas, anti-poaching measures, and captive breeding programs, are vital for preventing extinction. However, even when these efforts successfully boost population numbers, they can't restore genetic diversity that has already been lost. The loss of a unique gene variant can take thousands of years of evolution to recover through a lucky mutation. In a recent paper published in Nature Reviews Biodiversity, an international team of geneticists and wildlife biologists argues that the survival of some species will depend on gene editing, in addition to traditional conservation actions. These advanced genetic tools, already revolutionizing agriculture and medicine, can enhance endangered species by reintroducing genetic diversity that has been lost. Genetic engineering is not new. Plant breeders have used it for decades to develop crops with traits that boost disease resistance and drought tolerance. Around 13.5% of the world's arable land grows genetically modified crops. Gene editing tools like CRISPR are also being used in de-extinction projects that aim to recreate extinct animals. The Dallas-based company Colossal Biosciences has made headlines for its efforts to bring back the woolly mammoth, dodo, and dire wolf. In de-extinction, the DNA of a living relative species is edited to approximate the extinct species' most charismatic traits. For example, to resurrect a woolly mammoth, Colossal's researchers plan to splice mammoth genes, recovered from ancient remains, into the genome of the Asian elephant to produce a cold-hardy, hairy elephant-mammoth hybrid. Colossal recently engineered grey wolf pups with 20 gene edits from the extinct dire wolf's DNA. The Jurassic Park-style revival of long-gone creatures has attracted considerable attention and funding, accelerating the development of genome engineering techniques. These same tools can be used for the conservation of existing and endangered species. If we can edit a mouse to have mammoth hair, or edit a wolf to resemble a dire wolf, why not edit an endangered bird's genome to make it more resilient to disease and climate change? Museum specimens hold a treasure trove of genetic information. Using DNA from historical specimens, scientists can identify important genetic variants that a species has lost. Many museums hold century-old skins, bones, or seeds, a genomic time capsule of past diversity. With genome editing, it is possible to reintroduce these lost variants into the wild gene pool. By restoring genetic variation, species can be fortified against emerging diseases and environmental change. A sharp decline in population numbers is called a bottleneck. During a bottleneck, inbreeding and genetic drift lead to the random loss of genetic diversity. Harmful mutations can also increase in frequency. Such genomic erosion compromises the health of individuals and can make populations more prone to extinction. If we can pinpoint a particularly damaging mutation that has become widespread in the population or a variant that has been lost, we could replace it in a few individuals using gene editing. Aided by natural selection, the healthy variant would gradually spread in the population. And this is the part most people miss. If a threatened species lacks genes that it desperately needs to survive new conditions, why not borrow them from a close relative that already has those traits? Known as facilitated adaptation, this could help wildlife cope with threats such as climate change. In agriculture, such cross-species gene transfers are routine. Tomatoes have been engineered with a mustard plant gene to tolerate cold, and chestnut trees got a wheat gene for disease resistance. There is no reason why such techniques cannot be expanded to animals. These genetic interventions can complement, but never replace, traditional conservation measures. Habitat protection, control of invasive predators, captive breeding programs, and other on-the-ground actions remain absolutely necessary. Importantly, gene editing only makes sense if the target population has recovered in numbers enough, often through conservation, to allow natural selection to do its job. Gene-edited animals or plants wouldn't have a chance if released into a barren habitat or a poaching hotspot. Genomic tools can give an extra edge to species that are already being saved from immediate threats, equipping them for adaptive evolution in the future. Climate zones are shifting, new diseases are spreading, and once isolated populations are cut off in small fragments of habitat. Without intervention, even intensive habitat management might not prevent a wave of extinctions. However, a strategy of gene editing also comes with significant risks and unknowns. One technical concern is off-target effects. CRISPR and other gene editing techniques might make unintended DNA changes in addition to the intended edit. In other words, you attempt to insert a disease resistance gene, but accidentally disrupt another gene in the process. Similarly, a gene may have more than one function, which is known as pleiotropy. Especially in less well-studied species, we may not be aware of all those functions or pleiotropic effects. Regulatory inertia and public skepticism may also present big obstacles. These issues have historically limited the rollout of genetically modified, GM, organisms, particularly in agriculture. There are also evolutionary and ecological uncertainties. A deliberate gene edit might have knock-on effects on how the species evolves over time. For instance, if one individual is given a highly beneficial gene that spreads rapidly, it could replace all the other gene variants at that location in the genome, the full complement of DNA in the organism cell. This is known as a selective sweep, and it inadvertently reduces the genetic diversity in that region of the genome. Some critics argue that the narrative of a genetic quick fix could distract from the root causes of biodiversity loss. If people believe we can simply edit a species to save it, will that undermine the urgency to protect habitats or cut carbon emissions? Portraying extinction as reversible might seed false hope and reduce the motivation for tough environmental action. Conservation efforts, strong environmental policies, and legal protections remain indispensable. So do habitat restoration, climate action, and reducing the impact made on the environment by humans. Nevertheless, genome engineering is a new tool in the conservation toolbox. It's one that, given the right assistance and environmental encouragement, can help save species from extinction. Get your news from actual experts, straight to your inbox. Sign up to our daily newsletter to receive all the Conversation UK's latest coverage of news and research, from politics and business to the arts and sciences. But before you go, we'd love to hear your thoughts. Do you think gene editing is the key to saving endangered species, or do you have reservations about its potential risks? Share your opinions in the comments below.